So here's the genome annotation PowerPoint presentation. So this will be mostly lecture, but I will have your participation. And whenever it says pick a colored card, I'll either build a poll or you guys just answer the question in the chat. So the poll is anonymous. If you're shy, maybe that's how I'll get you to participate. All right, so you're going to learn today about the overall structure of eukaryotes and prokaryotes. You may not know what this means, but EU is not like EU. That's what my daughter says. But like EU is like true. And then this is a true karyon is like a kernel. And then pro is a prototype of like a kernel. Meaning, in this case, the kernel is whenever people look at the cells, they're like, is there a distinct part of the cell that it looks like kind of like a pit? And that's the nucleus. So a prokaryote, the nucleus is not as defined as an eukaryote. So the eukaryote has a membrane that envelops the nucleus. And the prokaryote, the DNA is kind of bunched up, kind of dissolved in the cytoplasm. I don't know how dissolved, but it's not as distinct as an eukaryote. So their genomes have some key differences. So this will actually be the very last slide. But a lot of the class will be on genome annotation, because this is the lecture where I hope you learn what a gene is. And in an exam, I ask you, what is a gene? And I'm looking for the physical definition of it. And that's what you're going to learn today. So what should you learn from the lecture? Whenever somebody says genome annotation, you know, if you are in a conference or you're reading something, they say the genomes, there is this group does annotation. I want you to know what that means. But you could think about it as you have a book and there's some passage of the book that you like, and you highlight that passage, and you not only you highlight, so that would be some level of annotation already. Maybe you want to write some notes to yourself on why you highlighted that. So that would be the genome annotation. Is you have the book, which are the A, T, C's, and G's, and then you want to say, oh, this chunk of the book is meaningful. I'm going to make that part bold, or I'm going to write this sequence encodes the gene that does whatever. So that's what the genome annotation is putting information on top of information. So this is a biggie, you know, what is a gene? And you might be thinking, oh, blue eye versus brown eye. Okay, those are alleles, but the gene would be a gene that encodes eye color, and it's not as easy as we make it seem like when you're teaching genetics. It's just like an introductory way of making people think, oh, okay, there are those variants for that gene. The variants are called alleles. This you have probably not seen unless you've taken genetics. I don't know if you see it in cell bio, but something you know here is 5' and 3', and you're like, ooh, I've seen that before. And then I'm like, yeah, you have. And I know I make kind of a big deal out of it because it matters. It's the orientation of DNA, right? The 5' UTR is the beginning of the DNA sequence, and the 3' UTR is the end as we see it, because DNA grows on the 3' end. So if we are making a word grow, we're adding letters to the right side of it, unless you're Arabic, in which case you write the other way. But the way we write DNA, we call it the 5' because of the 5' carbon, and 3' because it's to the 3' carbon that new nucleotides are added. So like, okay, I know the 5' and UTR and 3'. Dr. T, just tell me what UTR stands for. So this is the untranslated region, okay? That is not for transcription, it's for translation. So transcription is a process where DNA is being read by an RNA polymerase and is being transcribed to mRNA. So then you have an mRNA molecule. So that's transcription. But this T here is for translation. That is when the mRNA is being read, and then there's by a ribosome, and our mRNA is going in, proteins coming out, not because the ribosome made some magic and transformed that same molecule into something else, but it's reading one molecule and making another molecule. So the translation part is reading RNA, making protein. And the UTR means there is some part of RNA that does not actually become protein in the end. And there is some of it in the 5' end of the mRNA, some of it on the 3' end of mRNA. So you're going to learn all these concepts more in the exercise that I'm going to run with you, like on paper, staring at sequence. And you're going to see it again when we run a gene predictor software. All right, so, and this is more toward the end. What is the difference between, whenever I think of prokaryotic genome, I have it in my head, it immediately pops up, how is it different from a eukaryotic one? And, okay, isn't that as the same? And okay, which cues we use to predict a gene, that's, you're also going to learn that from the paper exercise. And how do we assign gene function? We can't really do that by just looking at a DNA sequence and knowing what it does. We have to compare that DNA sequence to some other DNA sequence that somebody already knows, and then we can infer, or we can break that gene, and then we know what it does. So I'm going to stop sharing this for a moment, because my daughter was making cookies the other day, and she wanted the simplest cookies, and there was, I don't know, I don't make cookies, but it's like she made almond flour, sugar, and I don't remember what else. I don't know if it was butter or what, whatever it was. And she didn't put, or she did, she put baking soda. I don't know. But I remember thinking, how would she find out what the baking soda is for? So, you know, we had this discussion, because she's like, oh, what are these ingredients? What do they do to my cookie? And I'm like, how would you do that experiment? So very proud. She's like, I'll make some with baking soda and some without, and see what happens. And that's how I'm going to infer the role of the baking soda. And I'm like, yes. So that's what we do with genes, right? So we want to see, there's this DNA fragment. I don't know what it does, but how about if I omit it? So there are laboratory techniques that you can do to break that, which would be analogous to removing not adding the baking soda. We can't not add. We could add more of it. That's the other one that sometimes you can do. So we can, let's say you cannot omit the baking soda, otherwise the cookie dies, or that would be with the living being. Maybe you cannot omit that. Maybe you're like, what do ribosomes do? I'm going to remove them. And the thing dies, and you're like, okay, dies, but I don't know what it does. So what you could do is add more of it. So those are the laboratory techniques to find what a DNA sequence does. Is we either break it, we remove it, or we add more copies of it. Got it? So those are the key points of the lecture today.